I was selected to give this talk because I have, as Ellen mentioned, 2 young male microbes that regularly consume the contents of my refrigerator. So I am familiar with the process. So Kathleen Jennings, I'm a scientific field responder with OSPR, and I'm part of the group that includes Josh, Craig Hafner, Mike Schomer, who you've all met during our field exercises and other presentations. And we're tasked with arriving on scene. We're first responders, so we arrive on scene and making determinations about what cleanup endpoints are going to be the most, what cleanup methods and endpoints are going to be the most appropriate during a response. The use of applied response technologies, in particular bioremediation, is a little bit different because, as Ellen mentioned, we don't use it as an initial response tool typically. It's usually used in combination with other response technologies. And oftentimes it's not initiated in the early hours of a spill. There's a delay, and then it becomes a more feasible option. So bioremediation has been around for 20, 25 years, used as in these types of applications or for hazardous waste site cleanups. So it's an effective tool, but there's some limitations to it, which I'll talk about. Do I shoot the screen? Yes. Have you seen this diagram about oil weathering? I'm putting it up there as a reminder that collectively these processes make up oil weathering, and depending on the product that you start with, some of these more than others will be at play. So if you have a gasoline spill, evaporation occurs very quickly. So we don't actually do active cleanup of gasoline spills for the most part. Cleaning it up is very hazardous. Most of it will go away pretty quickly within the space of hours to days, depending on the volume that's spilled. So as you move through these weathering processes, some of them take a little bit longer. So you have dispersion, dissolution, evaporation kind of occurring pretty quickly after the release. Emulsification, which you remember is the mixing of oil with water, like this is shown here. That usually doesn't happen right away until there's more turbulence and mixing. Adsorption to sediments is something that takes a little bit longer. Photooxidation is a breakdown of the petroleum from sunlight. And biodegradation is one of the slowest naturally occurring processes, and it can take anywhere from weeks to months, years, depending on the conditions. So biodegradation, is important to understand that biodegradation is a natural process. It's going to happen whether we do anything or not. And so in every system on Earth, there's some type of microorganisms that are present at those sites. Primarily bacteria and fungi, other organisms as well, though, will break down organic molecules, of which petroleum hydrocarbons are considered organic molecules, into other substances, and ultimately into carbon dioxide and water. They break it down, they degrade it to the point where it's innocuous. Bioremediation is taking advantage of the natural biodegradation process and trying to speed it up by adding things to the environment that has these petroleum hydrocarbons or other contaminants and enhancing it. So you can add fertilizers or other compounds. You can manipulate site conditions, and the idea is to speed up and accelerate the natural biodegradation process. So that's the theory of bioremediation. So this is just a little bit on bacteria. Bacteria are one of the primary degraders of petroleum. So there are different types of bacteria. These are not the kind that cause, staph infections. These are bacteria that are hydrocarbon degrading bacteria in the context of what I'm talking about. And they're different types. Some of them are aerobic. They need oxygen to do the degradation. There's some that are anaerobic, but the process is slower. They can actually degrade without oxygen, but the process is slower. So whenever we can have aerobic conditions with oxygen present, that facilitates the bioremediation. There are also bacteria that use different energy sources. So the heterotrophics, the term is other feeding. So they degrade other compounds to get energy for their biological processes. Autotrophs are self-feeding. They oxidize inorganic compounds and create organic compounds out of it. So they grow and do their thing that way. So the main point of this, if this is a little bit biological, is we like oxygen and we like organisms that consume organic compounds, okay? So what do hydrocarbon degrading microbes need? Food, primarily. So carbon, a good source of carbon is in petroleum hydrocarbons. Hydrocarbon, carbon with hydrogen. And they also need nutrients. So their soil, you probably are familiar with agricultural theory where if you plant legumes or peas, it adds nitrogen to the soil primarily, and it provides this nutrient, this extra nutrient that is needed. Well, microbes need nutrients like nitrogen, phosphorus, and sulfur. So they need a food source. They need oxygen. I mentioned aerobic conditions are more ideal. Yeah, I guess I can mention this. So oxygen, under aerobic conditions, there's a range of oxygen levels within soil that are ideal. So aerobic conditions are 2% of 2% oxygen gas within the pore spaces in soil, and I'll explain some of this later, or 0.4 milligrams per liter if there's water in the spaces between soil particles. A moist soil, it can't be totally saturated. It can't be totally wet with all the spaces in between the particles filled with water. Ideally, it's about 50 to 70% of the water that the soil would hold, and that's ideal for bioremediation. Temperature warmer is better. And a range of about 0 degrees Celsius to about 40 degrees Celsius, which is about 100 degrees Fahrenheit, which is a number we're more familiar. So pretty warm, it'll still work. Below 0 degrees, not so well. And they need a substrate, a place to live. So this diagram just shows it's not like Pac-Man eating the oil. These microbes actually secrete enzymes that break down the outer edge of the oil and degrade it into different compounds, smaller compounds like carbon dioxide and water. Is everybody kind of with me? This is some of your biology, high school biology. So the way that we conduct bioremediation is we know what the microbes need, and we work to identify what is limited in the environment that we're thinking about doing bioremediation. So are there not enough nutrients? Well, maybe adding nutrients will provide more bacterial growth and petroleum consumption. Is it oxygen they need? There are things we can do to increase the oxygen levels. Are the soils saturated? We can do things to get rid of some of the water between the soil particles and bring more air and gas in. Is it cold? Not so much we can do about that. You can't really do bioremediation in permafrost in the Arctic because you can't control things like heat. So some of these are a little bit easier to control and work with and manipulate than others. And microbes, Ellen mentioned, but one of the things about microbes is they're everywhere. They're ubiquitous in the environment. Petroleum degrading microbes are also ubiquitous in the environment. They find them everywhere. They found them a mile deep, at very high pressures in the Gulf where they were releasing oil from the wellhead. So they're everywhere. You also, like everything else, have to consider the physical and chemical nature of the oil and the environmental medium. This works better with heavier oils. Yeah, a little bit about that. I mean, the main thing is when you introduce, there have been a lot of situations where organisms were introduced to solve a problem. Right. And, the larger scale stuff, like mongooses to Hawaii and eating all the native birds, it's the same idea. So you don't know all the consequences of introducing microbes. The bigger thing is that if you introduce microbes, you're trying to enhance bioremediation, typically they're just outcompeted by the native microbes that are there, that are acclimated to the conditions at that site, and they just outcompete them. So it's just not very effective with bioremediation.
We're not going to end up with giant worms.
We hope not. So there are 4 primary bioremediation strategies. And some of these, and actually you have handouts for this talk in your notebook, so this is probably a good time to mention. Alan Mearns with NOAA was one of the people that's been working on bioremediation for 20 years. He gives a rundown of these different types of bioremediation strategies. His article is wonderful. He also gives some historical perspective. So that's one of your handouts in the book. So I would take a look at that. So biostimulation, adding nutrients or other chemicals to stimulate the natural process. Bioaugmentation, which is adding microbes, which we just talked about. Can you guys hear me in the back? Okay, good. Bioturbation is where you colonize the sediment or soil with plants and animals, mix things up, increase pore space, increase aeration. And physical manipulation is doing the same thing, but doing it mechanically. Okay, so these are just the overall theories, and I'll talk about them a little bit more detail. Ellen showed that dispersants can be considered a bioremediant because what it does is it breaks the surface slick into smaller droplets. It increases oil surface area where the microbes can get in, and the smaller droplets are more bioavailable to the organisms that are smaller. So, and I mentioned that the ultimate process is degradation into carbon dioxide and water. And I won't go into too much detail, but essentially, the smaller droplets have the surface area is more available for larger droplets or big slicks. They work on the outside edge, so there's always going to be oil remaining. When you break it down small to organisms of around this size, it's easier. They're more bioavailable. The organisms can break them down easier. So that's the theory. And Ellen mentioned this too, but the decision they had to make, and I think Jordan will talk more about environmental trade-offs. You don't get something for nothing. So they had to decide between chemical biostimulation, to enhance the degradation of the particles by making them smaller with dispersants, versus the possible toxicity of adding that much dispersant, 1.8 million gallons of dispersant in the Gulf to break them down. So there are always trade-offs, no matter what you do. And so we know in the Gulf of Mexico that there's already an established, diverse petroleum degrading microbe community. There's oil in this environment naturally, so there are microbes that are adapted to those conditions. Lots of hydrocarbon degrading bacteria. This was actually one that they discovered as part of the research they were doing, and we know that usually bioremediation is a longer-term tool. The way they used it, though, they were able to use it at the wellhead, so it was actually kind of a primary response tool in that situation. But they couldn't. The bugs couldn't keep up with the oil at that rate. They weren't able to degrade it fast enough to get rid of all of it before it reached the surface. So that's why we had impacts resulting from the spill. And as I mentioned, they even found microbes down 1 mile deep at the wellhead, where the temperatures are low, the pressures are high, and they were there. They had adapted to those conditions. And so another thing about biodegradation in general is that it's nonlinear. It starts out really high. The microbes bloom, they eat, and then the rate's lower. And I'll explain a little bit about that as we get a little further in. So now I'm totally switching gears because the case study I'm going to present is one actually you guys from Solano County are probably going to be interested in. This is for Soon Marsh. And in order to understand kind of the context for the context for the case study, I need to just give a little primer on soil science. So within the soil matrix or sediment matrix, there's a framework of mineral matter. And this is stuff like sand particles, silt particles, clay particles, usually a mix, but it represents about 45% of the whole soil matrix, okay? Between those particles, you have air, and in certain depths of the soil, the air might be greater or less. You have water in different degrees, and organic matter. And this stuff is all between this framework based on the particles. Okay? I think I just covered that. So, hold on one sec. No, we need to go back. My diagrams got lost. See, I guess there's like smudges. Well, my screen, it looks great. So soil particle size helps facilitate bioremediation depending on how the soil is organized at a site. So this is, I use this, there's a little mnemonic I use, and that is, it is clay, silt, sand, gravel, cobble, boulder in a size range, similar to what we did yesterday during the SCAD exercise. And this, a lot of times, we need to know this when we go out to do soil bioremediation. An easy way to remember it, I came up with this one, I like it. Crickets sing songs getting comfortably baked. So if it's easier to remember it that way, which it is for me, but it's the size particles. Clay is the smallest particle size, and you can see clay particles are kind of flattened. They're kind of flattened like a fried egg on its, in profile. And so when they come together, they kind of overlap, and so there's not much space between the particles. They lay pretty flat, and they can provide, they don't mix, there's not a lot of water or air that gets between those particles, and that's something to know. Silt is a little bit larger particles, but there's still not that much space between the particles. It's too bad you can't see my pictures. Sand, the particles are a little bit bigger. There's a little bit more space between the particles. When you look at things like, cobble and boulder, there's a lot of space between the particles, particularly because they're not round particles. They're edges and things sticking out, and so it increases the space. You have a question, Jeff?
It's in the book.
Oh, good. Did it show up in the book?
Yeah, it does.
Oh, good. Okay. Makes me feel better. Okay, so you need to just understand that, because I'll be talking more about this with the case study. And keep asking questions. This is a lot of information thrown at you. So this is what the soil matrix kind of looks like if you're doing a cross section of the soil. So you have the surface of the soil. Usually you have an unsaturated zone or a vadose zone. You have particles of different sizes, and then within the spaces, which are called pore spaces, you have organic matter, air, and water in different combinations at different levels. I mentioned clay lens. If you have a patch of clay, even in your unsaturated zone, you can have a perched aquifer or an aquitard where water stays above it. Because remember, clay is not permeable. You know this from working with clay or building stuff. Clay will, hold water. It doesn't. That's why you have to mix it with other stuff in your gardens when you're doing gardening. So the water table is the line, and this you probably can see this better in your handouts too. The water table, there's actually a line here, and the water table is a line where the space between the soils is filled with water, which means there's less air, or no air in this point. There's some organic matter, but there's no air below the water table in the saturated zone. And your bedrock usually forms the base of your soil profile. And usually when you have a groundwater, an aquifer, a water storage unit in the soil, there's a bedrock layer, and the water accumulates above that. So I think that will help us get to the next step. So we have that kind of framework and soil matrix and pore space stuff done. There's also all these organisms living in soil. Soil is not a dead medium. It's very much alive. We have organic matter from plants and roots and decaying stuff. It's waste and dead bodies from all different organisms in the soil. We have the fungi and the bacteria that are decomposing things. They're worms and insects. All this stuff is occurring in the soil simultaneously and doing its thing. So there's all this stuff going on. It's a dynamic system, even though it seems like a dead system. So that's what you need to understand about soil. So now I'm going to get back to the bioremediation techniques, because this will help as we walk through. So biostimulation, which is the enhancement of bioremediation through addition of nutrients or other substances. When you add nitrogen and phosphorus to pore waters of oil-contaminated soils, so remember the pore waters, this has been tested and it's shown to be effective. So what happens is, and this is the process, this is the theory, you have naturally occurring petroleum degrading microbes in a soil, and you add in nutrients that they need, and you increase the populations of these microbes, and it happens pretty quickly and dramatically. You provide an extra food source, and their response to that extra food source is to bloom, is to increase their numbers substantially. And so once they consume all the nutrients that you've added, then they're looking for food, and we have an alternate source here, which is petroleum hydrocarbons, all organic substances. So that's the theory. And then once the supply of the petroleum is gone, those hydrocarbons, then the microbes start to die off, because it's getting harder and harder for them to find a food source. So there are different theories, but you can then spike with nutrients again to increase the bloom, or you can kind of let things do their thing and wait. There are a couple different theories of following up with that. So that's the idea of biostimulation. Bioaugmentation, which is what Ellen mentioned, is where you add microbes. And when they're in combination with nutrients, you can see some effectiveness, but it's probably more due to the nutrients than it is to the microbes they're adding. Fairly poor results with lab and field tests. If you add them alone, they don't typically increase biodegradation or and I mentioned that this was the reason, in response to your question, Bob, that they occur. Hydrocarbon degrading bacteria are in all soils. They're in diverse communities. They have different places within the environment that they survive, and they will outcompete what you throw in because they're adapted to those site conditions. Bioturbation, and Alan Mern's paper talks about this. Bioturbation is where you use plant roots or soil and sediment animals that break up that soil, that increase that pore space, that increase aeration, water circulation. They even use plants to uptake soluble chemicals. Those are things not like oil, but things that dissolve in water. They use them for like chlorinated solvents. They will use plants to pull them up out of the soil, and then they'll dispose of the plants that have these materials as hazardous waste. So bioturbation. And then physical and mechanical manipulation. So if you have none of these chemicals, but you really want to enhance bioremediation, you can till the soil. And this will increase the pore space between the particles, and it'll increase aeration, add in oxygen, and mixing. And so this by itself will enhance. You can also do things like if you have a shoreline that gets oiled, if you do shoreline washing, and this is usually with high pressure, it'll decrease the oil droplet size and increase aeration and mixing. So there are different methods to get to the same place, but these are all things that we can do to enhance bioremediation. And Ellen talked quite a bit about this, about the oil spill cleanup agents like dispersants or bioremediation products. The benefit of using agricultural fertilizers is they're not a regulated, they don't have to be on the big list of oil spill cleanup agents that you can use. So a lot of bioremediation projects focus just on agricultural fertilizers. It's still a good idea to work through the regional response team and get their approval and involve them in the process, but it's not required in the same way the others are. And so one of the things is you don't really even need to worry about what you're going to use until you determine if it's even feasible given the conditions of the site. So these are the things you need to determine in deciding whether active bioremediation is going to work at a site. Are the background nutrient levels low? If they're not low, adding more is probably not going to have much of an effect. There aren't enough. There's enough there currently, and the organisms are that matches the population of organisms. Are the environmental conditions appropriate? Are the right microbes, the hydrocarbon degrading microbes there? Is the temperature within that range that will allow this to work effectively? Is there oxygen present in the air spaces between the soil particles or in the water between the soil particles? And this is, I think, where I mentioned it was 2% oxygen in the soil gas in the soil pore spaces, and 0.4 milligrams per liter of oxygen in the water within the pore spaces. And then we talked about moisture being 50 to 70% of the saturated conditions. Is there a feasible nutrient delivery system? So if you're going to add compounds, you need a way to be able to add them effectively. If it's a small area and you need to helicopter in your substances, it's probably going to be a lot more difficult to kind of get this initiated, do the monitoring and all of this. So you need to have the right setting to be able to do this effectively. And then is long-term treatment an option? Because remember, the weathering process, biodegradation was one of the slow ones. So you need to allow enough time for this to take place. Okay, so let me point out a couple key features here. Josh, you're doing my time, right? Yeah. Okay, thanks. Okay, so we're somewhere down here sitting in this building. Sassoon Marsh is that when you're up on the freeway and you look off to the east, that's Sassoon Marsh, that big contiguous marsh. This is I-680 here, and this is Interstate 80 here at Cordelia. So the site where this occurred is right here. And if you remember this is Sassoon Slough, this little waterway here. This line here is a high-speed railway. Okay, gosh, that's hard to see. So key features. So again, this is a closer up shot of the site. This is the railroad line here. This X is where a pipeline was ruptured. It was a catastrophic release of about 100,000 gallons of diesel. This happened in 2004. And this primary area here, which is called Division A, this area here was the most heavily contaminated. It's the closest to where the release occurred. These other areas that got contaminated are because the oil came out of a tide gate and moved into these tidal sloughs that move through the marsh. This is a managed marsh. It's a diked marsh, so you actually have some degree of control over water movement by opening and closing tide gates. So it's an ideal situation. It allowed us to control things like soil moisture at the site. So these are the tidal sloughs. If the Sassoon Resource Conservation District did not close those tide gates sooner, the impacts to the outer areas would have been much heavier. This is about 220 acres. This is about 10 acres in Division A. I'll show you right now here. So again, railroad tracks, release site. This is Division A right here, and there's some numbers here. The top half, about this much up, was the most heavily contaminated, the soil, and I'll explain how that occurred. This area wasn't contaminated, and as I mentioned, this other area was primarily in and around the sloughs that work through the site. So this arrow indicates overland flow. So when the rupture occurred, there was a blowout, and this product was released very quickly, and there was so much of it coming out so quickly because the pipelines are under pressure that it ran across the surface of the soil just in a line straight out to the tide gate, which is right here. And this is the one that was closed by the Resource Conservation District pretty early on. So the main surface soil contamination was right in this overland flow path, and I'll show you what that looks like. Oh, I want to say one other thing. So early on in the response, trying to decide what the appropriate response methods were, looking at the top half of this part is 20 acres, so the top half that got heavily oiled, so you figure 10 acres, there's about 3 feet of soil before you hit groundwater in that area. And I did a little quick calculation, so it worked out to about 50,000 cubic yards of contaminated soil. And if you look at a truck holding a 20-yard storage bin, it's like 2,500 truckloads of soil that you need to get in and out in an area that is very unsafe. There's a little gravel road 10 feet away from this high-speed rail line, and you come in on it, and up here you actually have to cross the railroad and do it quickly. So this site was, this wasn't a great option, doing excavation and soil removal. It's costly and it's dangerous, and when you remove that much soil, where do you find replacement soil? The soil is very specific to this type of marsh setting. 10 minutes, okay. And so that was one of the issues that came up, and so we decided that excavation removal wasn't a good option. They looked at in situ burning, which Ellen talked about. They actually went so far as to get approval to burn the oil in this marsh area from the Air Board and all the agencies, but they decided not to do it because of issues related to people driving up 680, riding the rail lines, the smoke that would be generated. Controlled burning is one of the management techniques they use in this marsh regularly. This would have been bigger and blacker and more noticeable, so they opted not to do that because of concerns for safety. So within Division A, these were the activities we used in concert. So pipeline soil excavation removal of the heaviest impacted soil. They did some trenching and free product recovery. Then we went to bioremediation, and they did monitoring for a long time. So this is what the pipeline looked like. It's a 14-inch refined product line. So it has gas or diesel or jet fuel running, and when it ruptured, it had diesel in it. So 100,000 gallons of diesel. This is the pipeline trench here, and this is, again, the 14-inch line. They had to excavate 1,000 feet to repair the pipeline. So this is the product, what it looked like here. Here's a skimmer. You learned about some of this yesterday. There's the pipeline replacement piece. When they replaced it, and the 8-inch line behind it that goes to Travis Air Force Base, which is a very important one too, they added Teflon coating because this is a corrosive environment. The water table, as I mentioned, is only 3 feet down, and so the salt water really corrodes the pipe. So they added this Teflon tape as a precaution. This is the overland flow path. Here's a back truck right here. Here's the railroad tracks. There's a release location. Here's where it ran heavily out to the tide gate, right across. This is really the only surface soil that got contaminated. But what we found out is we closed the tide gate. This was in April. This happened in April, or late April, end of April. So as we closed the site, there was no more water coming into the site. These desiccation cracks formed, and when we looked a little closer, this is a crack, and we dug a hole. There was 4 inches of standing product in the bottom of that little hole. And it was more like surface flow than flow through the soil, which is very slow and doesn't flow. Like this is more like a little creeks of oil. that formed in the desiccation cracks. So we followed those cracks and we dug a bunch of trenches and we got as much oil as we could using pumps. We couldn't get back trucks down there because the soil was too wet. And we removed as much as we could to the point where we were mostly getting mostly getting water. Then it's like, what do we do now? Ask ourselves these questions. Harry Allen was the federal on-scene coordinator for this spill. Harry Allen IV. Harry Allen III is a specialist in bioremediation, so I'm sure that had something to do with it. So these were the questions that came up at this point. Our background nutrient levels low. So after testing, they figured out, yeah, they're low enough that if we add something will happen. Microbes are there that we need, the hydrocarbon degrading microbes. The temperature was good. It was, less than 100 degrees, which is the upper end of the range. There was adequate oxygen, which could be enhanced a little bit. Moisture was a problem because the soils were fairly saturated. Could we do a feasible nutrient delivery system? Yes, and is long-term treatment an option? We had from, say, May 1st until October 1st, which is when all the tide gates are supposed to be open for duck hunting to flood all these lands. Did I mention this was a duck flood? I'm not sure I did. So they did a bunch of testing, soil testing, determined that everything was appropriate to be able to do bioremediation. The first thing they did is they scraped the soil, the surface soils. Remember, the only place that was heavily oiled on the surface was the overland flow path. They scraped the soils and they stockpiled them right here. Does anyone want to venture a guess why? It provided the seed bank for the restoration of the site once they were done. Native seeds, native plants, all the native stuff, and they could revegetate using the original seed bank because the surface soils were clean. The other thing we had to do before we scraped, there's a listed species out here, salt marsh harvest mount. And so we trapped out what we could and relocate them to another area of the marsh. So this is the soil, this is the nutrient delivery system right here, Matt. We had to aerate the soils to dry them out a little bit, so we created windrows with a backhoe and got the site ready. This is what it looked like before. Craig showed this diagram earlier. These yellows are water monitoring wells. This is a levee road that didn't need to be there, and the property owner, who the duck clubs here, wanted it gone. So I'm pointing out these key features. Sure. This is what it looked like after with the windrows formed to dry out the soil. There's the stockpile. Here's where we did the nutrient addition. 2 Baker tanks with water, 1 tank with nutrients, and some sort of a mixing system. This is the hand-neat thing that they shipped out here to spray the nutrients in the soil. It got stuck in the mud within an hour. They ordered new tires for it. Those got stuck too, so they ended up dragging it behind one of these with the big tires. That's funny. And so after they started doing this, then they started doing weekly monitoring to see whether things were working after they added the nutrients. And this is what they found. Soil and nitrogen, remember I talked about nutrients being one of the limiting factors? So they were about this range. They added them on the 8th. And when you see nutrients decreasing in soil, that lets you know that something's consuming it or using it. This was a weird anomaly because on the 27th, they added more nutrients, but things still continue to go down, which I'll explain in a minute. Biomarkers, this is something they use to determine if it's bioremediation occurring, not other processes like evaporation. They did the biomarker ratio, so they look at something that's easy to eat versus something that's hard for the microbes to eat, and they compare them. And the ratio went down, so the easy to eat stuff went down preferentially over the hard to eat stuff. And again here, there's this weird thing happening around this time, which I'm going to explain. Heptadecane is the first part of the ratio. These are straight chain alkanes. Every point has a carbon molecule. This is like cotton candy to these microbes. They just start on what, it breaks down easily. There's no energy. It melts in your mouth. It's an easy thing. This is what they eat preferentially. The thing that it's compared to is Pristane, which, remember I mentioned this was diesel. The carbon range for diesel is 10 to 20 carbons. So this is within the diesel range. This is within the diesel range. This is called a branch chain alkane. So there are more connections. It's a little bit harder. It's like beef jerky. You gnaw on it, but you can still eat it. Okay? And versus something like this. This is a ringed alkene. So these things indicate double bonds. This is really hard to eat. You have to put a lot of energy into boiling and getting an artichoke ready, and then you get only a little bit of food from it. So that's simplification of what this is about. But when you look at the ratios and use these things, we were able to determine pretty definitively that bioremediation was occurring. These are the results. These are TPH diesel concentrations, and they went down dramatically. This is more dramatic than actually occurred because when they mixed all the soil to get the windrows, set up, they homogenized the soil. So they mixed in the areas of high contamination with low. So some of that downturn is represented by that. Remember I mentioned the 29th of July? On the 27th, we added nutrients. On the 28th, the people that were working on the pipeline trench decided that they had a lot of extra dirty soil, and they'd throw it into the bioremediation pot and just mix it in, and they didn't tell anybody. So we didn't know what was happening with the results until we saw some of this. That's why the concentration went up on the 29th instead of down. We did some sheen testing where we took soil samples and looked at whether sheen came up out of it. This is the qualitative endpoint. And finished off the bioremediation process. It's been monitored. So fast forward, this is 2007. Remember I mentioned that levee road, to get that levee road removed took about 4 years of Corps of Engineer permitting process. They recontoured, they had a plan, they recontoured the site to make it better habitat for the stuff that lives there, including the salt marsh harvest mouse that had to be trapped out. This is 2008. You can see they used the stockpile, things are starting to be revegetated. And this is 2012 that I got from Google Earth, which is why it's turned on its side. This is the site now, and you can see it's really filling in and becoming more what it should be as far as habitat. And that's it.